is pollen limitation and reproductive success of 5 South Puget Sound prairie plants. So I wanted to go into what pollen limitation means and how it's used. It's used to assess pollen distribution in plant communities, and it's based on what's known as the optimality theory, which is centered around the theory that plants living in their optimum environment are going to produce a maximum seed set being seed quality, seed quantity. And this is measured through pollen supplementation or hand pollination treatments, and then those are compared to open or natural pollination. And if hand pollination treatments produce higher seed set than open pollination, then pollen limitation is occurring. And pollen limitation can occur for several different reasons, mostly environmental disruptions such as habitat fragmentation or changes in pollinator networks. So my research questions surrounding this are, is pollen limitation occurring for any plant species? Does reproductive success vary by restoration site? And do native and non-native invasive plant species differ in reproductive success among sites? So this is a table of the 5 plants used in this study. And we have, the table shows the varying life histories, the floral morphology, native or non-native status, the phenology or the bloom time, and self-compatibility. And these are all different factors, different variables for each plant species, which makes them not comparable amongst each other. So the comparisons will be done within the species itself. And self-compatibility in a plant species means that they're capable of producing fertile seeds without external pollination. And so Plectridis conjusta is an annual native, and it is somewhat self-compatible, although a low rate. Lupinus lepidus is a perennial native, it's self-incompatible, and Eriophyllum linatum is a native perennial that is self-compatible, so that is something of note. Hypocris radicata is a perennial non-native, and it is self-incompatible, and Leucanthemum vulgare is the same, non-native, perennial, incompatible. Here we have the 6 different restoration sites in the South Puget Sound. They're all South Puget Sound prairie remnants. They're heavily fragmented sites, and they range in high or low quality prairie. So like high quality prairie would be defined as sustaining an established native plant community that can host native wildlife. And a lot of the high quality prairie sites are home to like reintroduction efforts of endangered species like Taylor's checker spot butterfly, while low quality prairie sites have undergone multiple land uses that have degraded the land and resulted in invasive populations forming monocultures. So those are the kind of the differences among these sites, and they kind of vary along that. Glacial heritage has the longest restoration history, over 20 years, and it's probably the highest quality prairie site. It's also the largest. Wolfhaven is the smallest in acreage, but it has very high quality prairie. It's home to Castanea lepisecta, threatened plant species. Tennelquot and Johnson Prairie are also high quality. Cavness Ranch is a conservation easement with multiple land uses still in place. And so it has areas of somewhat high quality, but it's predominantly very low quality. And Deschutes is the most recently acquired in 2015 by the Center of Natural Lands Management, and it is the lowest quality site. So the pollen supplementation experiment, this involves sampling individual inflorescences, and the way this was done was hand pollinating the inflorescences and then snooting them, which you can see in this photo is a nylon bag and it's tied around the inflorescence to exclude pollinators. And this was compared to open pollination treatments, which was just letting flowers be pollinated naturally, and then seeds were collected later. And then we already had self-compatibility data, so we already knew which plants were self-compatible. The data collection, so seed quantity was measured by a proportion of fertilized seeds, except for Leucanthemum vulgare, where just total seed set was counted because fertile and infertile seeds were indistinguishable. I also, for seed quality, calculated individual seed weight and then did a tetrazolium stain on Plectritis and Hypochorus. And the TZ stain is used to determine seed viability, similar to a germination study. And in the pictures here, you can see how I did that. The stain will turn the seed ovary pink if it's viable, and it will leave it kind of opaque white if it's non-viable. So the results for the pollen limitation were somewhat surprising. Treatment was not a significant factor in any models for any plant species. This is not what we expected, but it could mean that no pollen limitation is occurring for any of these plant species at any of these restoration sites. Or it could indicate issues with the sampling methodology. So in the literature, it cites that multiple hand pollination treatments are ideal throughout the phenology or bloom cycle of the plant. And in this study, only one hand pollination treatment was done before the snooting and was left. So that is something to change in future pollen limitation studies. Also, the IPU, or the Integrated Physiological Unit. So this is a morphological unit of study. So for the plants in this study, it was the inflorescence. But it could, for perennials, let's say, in the literature, it said the whole plant would actually be a better IPU of measurement. So that could be changed for the perennial plant species in the study. So those are some factors that could be changed in the future that might help with the pollen limitation methodology. So the results for reproductive success were the same measurements, seed quality, seed quantity. And here I have just a legend of the different site names and abbreviations I used. And for Plectritis conjusta, we have, what did I do? Fertilized seed, and, oh, I see what I did. I'm sorry. Okay, so Cavnus was significantly higher fertilized seed weight for Plectritis conjusta. And this is somewhat surprising because Cavnus was a low quality prairie site, but this could be due to, there was research done by Dunwoody et al. In 2014 that that native prairie annuals, such as Plectritis conjusta, can have reproductive output increases after burn and seeding treatments, which was done recently at Cavnus site. So this is just a one-time study, but if there was a long-term study, it could actually track the establishment rate or the long-term reproductive success. So that could be what Plectritis is experiencing at Cavnus. Lupinus lepidus, the significant data for this was the proportion of fertilized seeds was highest at Glacial Heritage, which makes sense because Glacial Heritage is the highest quality prairie site. So that was in line with what we expected to find. Eriophyllum linatum had no significant results for seed quality or seed quantity among the sites, and this is most likely due to its self-compatibility, but it could be, if it is studied again in the future, it could be helped by changing the IPU to the entire plant. Hypochorus, which is the non-native invasive plant in this study, had some mixed results. It was interesting, not what we expected either. As you can see for the proportion of viable seeds, also the letters denote a significant difference in means. So for the proportion of viable seeds, we have Hypochorus, actually the high quality sites like Glacial Heritage and Johnson Prairie and Chenalquat, and having a lower proportion of viable seeds at low quality sites like Cabnis and Deschutes. So this could be because Hypochorus is actually responding positively to restoration treatments at the high quality sites, even though the populations are reduced potentially. And we also have an interesting finding of a high quantity at Cabnis, high quantity of fertilized seeds, and a low proportion of viability. So that's another interesting finding where research would have to be done, site-specific data like plant species richness and different factors to further interpret why that's occurring, but it's interesting. Leucanthemum vulgare had the lowest seed weight at Chenalquat, and this was an interesting finding when compared to Johnson Prairie and Wolfhaven. This was interesting because it is specifically a targeted species in the Chenalquat restoration management plan. So potentially the restoration work at Chenalquat to remove leucanthemum is working. So reproductive success, restoration site was significant. So it wasn't exactly what I went into this looking for, but it is a really interesting finding. And this makes sense due to the heavily fragmented plant populations and the differing land use and restoration treatment histories. And different factors influencing plant reproduction are differing plant communities, differing pollination networks, soil characteristics. And this type of study could be applied to restoration practices to monitor reproductive success of native or non-native plant populations, assess different restoration practices, and also highlights the significant differences of these restoration sites. And future research that would be really interesting and complementary to this would be site pollinator networks and site plant species richness. Also doing a pollination limitation study with using different methodologies that I went over and suggested. And I just wanted to thank Sarah Hammond and Susan Waters, who I worked with a lot on this thesis, and Saunders Freed for providing the land management plans, and the Evergreen Science Support Center for letting me use the lab. And thanks. Any questions?